what I want to do is talk a little bit about a project that's coming along, and it's very much related to foodborne outbreak investigation and detection and traceability. And it's the 100K Genome Project, as we sort of like to talk about it. But as an introduction, I know you guys know a lot of this, but just to kind of get everybody on the same page of where we're sort of coming from. And you know the agricultural system is complicated, and it's a big interplay between the environment and health and safety and biosecurity. And we have all these new pressures put onto us as an agricultural entity in the U.S., along with imported foods. And now there's, you heard I think yesterday about FSMA, or the Food Modernization Safety Act. And it's allowing all sorts of new things to start occurring. And on top of that, the estimated population of the world will be approximately 9 billion people by 2050. So I'm old, but I'm not that old, and that's within my lifetime. So I'm not so sure how we're going to feed all these people when we have so many people in different parts of the world, and the vast majority of the growth is going to be in what's termed today the developing world, not in North America and not in Europe. And so now we have this whole new distribution chain, production paradigm to think about. And we're moving further and further away from the farm, so it's predicted that we'll have very large megacities in the future. So that means we're going to be packed in more closely. Food safety problems will continue to go up. And this isn't intended to be an extensive list of what's happening, but if you just look at 2013 and it's only April, mid of April, there's snow in Minneapolis, and there's a whole bunch of outbreaks that we've already had, all the way from pocket pets like hedgehogs, and it's Easter coming up, and there's already warnings about chicks having salmonella coming for Easter. And we've had pet food outbreaks already, and we've had vegetables and meat outbreaks as well, including turtles. And that just keeps going on and on. So if you really sort of look at what's the statistics for foodborne illness, FBI doesn't mean the guys in investigating crime. It means foodborne illness in this case. And so, you know, norovirus is still the largest proportion of what's going on as far as foodborne disease. And salmonella is still fairly high. But if you look at the deaths associated with those organisms, the paradigm shifts a bit. So despite the fact that Norwalk virus is very prevalent, deaths are fairly low. But when you start looking at Listeria and Salmonella, and now starting to be a bit more Campylobacter, then the paradigm shifts a little bit as far as what's important. And so I'll talk a little bit more specifically about Salmonella because it's such a hard problem. And many of you may have extensive experience with Salmonella already. But it's a hard problem to crack because it's so diverse. And the age groups in which the Salmonella continues to infect. And in a paper that we were involved with more recently, we can very much pinpoint the genetic susceptibility and the genetic traits that lead to hypervirulent Salmonella emerging both in animals and in the environment. And in this particular paper, we were working with a group where we were looking at cholera sus, which is transmitted primarily in sheep, and it becomes hypervirulent in close quarters with sheep. And now we have isolates that are super virulent as well, which means the infectious dose goes down to about 100 instead of about 10 to the 5. And it's natural evolution of what's going on in the population. And so just to remind you about the complexity of Salmonella and the diversity of the naming system, I mean, there's a huge number of serotypes, and the ones that are most commonly associated with foods, and animals specifically, are around 1,600 serotypes. And that's still a lot of bacteria to think about. And so there's about 50 or 60 most common that are really people think about on a week-to-week basis. So narrowing down from the 2,500 to around 60 serotypes is becoming a very important aspect of what's happening with outbreak investigation. And the whole point of this is that if we try and detect all 2,600-ish, it becomes a very big analytical challenge. And so as things have occurred in the regulatory space, we've tried to get rid of Salmonella broadly. And we've done an okay job of getting rid of Typhimurium. And so its prevalence tends to decrease. This is from 96 to 2007. But as Typhimurium goes down, then all these other serotypes tend to rise. which is highlighting the complexity of serotypes and the complexity of Salmonella, and that all Salmonella are not created equal. Despite the fact that serotype doesn't necessarily predict disease and it doesn't necessarily predict virulence, it does indicate the genetic diversity and the differences between the organisms, even though they're all called Salmonella. So one of the ways that we like to name bacteria is by using this 16S gene. It's one gene in the genome. It was discovered in the about 60s. A guy called Carl Woese said, let's take this gene, and it's slow evolving, so we'll name bacteria by this one gene. So when you make this a tree to align the similarities among Salmonella, they look really different by serotype if you use this single gene. But if you then take that and you align it by the whole genome, all of a sudden they look much more similar than we had thought they were before. And in my own work, I know that genetic evolution of one gene, that's why this tree starts spreading apart. That one single gene starts changing over time, much faster than the entire genome. And so the point of this is that some of the serotypes that we knew were similar by where they occur start clustering together. So we might be able to do something that's similar. And as we've started to sequence more and more of the genomes, then we start realizing that they're very different. And so the way to read this chart is everything that's red, got a red bar, is common among each of these isolates. And every place there's a black bar. It's new genes that the other isolate doesn't have, and we've just discovered by sequencing the genome. And so FDA has adopted very carefully sequencing genomes, or what's called next generation sequencing. And as we do this, we start discovering more and more genetic diversity among the organisms. And so this is particularly with Salmonella. And so every new isolate that we sequence, we find another new family of proteins and genes that we've not seen before. So at this point in genome sequencing, this is a bit disconcerting because we thought that we knew most of what was going on. And in fact, we know very, very little of what's going on the genome basis. And so as we continue to sequence more and more, then we can start seeing the genetic variation among the serotypes of Salmonella. And the way to read this flower plot is there's about 2,800 genes that they all have. And then each of these petals are a set of unique genes that are unique to that particular serotype. So the other thing to realize is that serotype is not at all predictive of genotype. So all Typhimurium are not just Typhimurium. And if you use the back system, you know that, because there's 17 to 20 different profiles in the back system just for Typhimurium. And Typhimurium has a huge amount of genetic diversity. So what's happening is the landscape is starting to shift from what serotype do you have to what genotype do you have. And I'll give an example of that in a little bit later. So because we really know so little about the microbial diversity from a genetics perspective, and we know that horizontal gene transfer is rampant among bacteria in stressful situations, which is what we have in food processing and also in animal growth, that we know we're starting to see genetic shifts due to rapid evolution. And there was a very nice paper published in 2012 discussing the evolution of Vibrio. And this paper was done specifically in the oceans off of California, but it's broadly applicable. And as you all know, Vibrio can survive very well in salt water, and it evolves quite a bit faster than what we had imagined. Some of you may have heard about this acid mine drainage community in California. There's 4 bacteria, the pH is 1.5, and the evolution of that community takes about 50 years. So that means a new microbe will enter that community and be stabilized in 50 years. But that's not the case in oceans or in Vibrio. Vibrio can evolve within a few weeks to a few months, and it's largely because of horizontal gene transfer and local stressful pressures to the microbe. And so they think it's stressful just like we think it's stressful when we throw acid and salt on them. And so the stress leads to these single nucleotide polymorphisms, where they exchange one base for another, which then changes the function, which then changes in some cases what we call it, because that 16S gene changes. And it starts mutating the DNA repair genes, and so pretty soon the genome starts to evolve very quickly because it can't repair its own DNA. And that's what happens in many, many microbes. And so you get these large segments of transfer, which leads to large genetic diversity very quickly, like new serotypes in Salmonella. And so this paper does a nice job of clearly explaining how and why this is going to occur, and then you get different lineages of bacteria. And so when we first started using PCR and genetics in the food arena, we thought that horizontal gene transfer was fairly simple. They only exchange DNA between near neighbors. And we know that's not true today. We know that it's very likely that you're going to get DNA exchange between different and diverse bacteria. So one clear example of this is E. coli 0157, which acquired the Shiga toxin from Shigella. Near neighbors, they both appear in the gut. The toxin genes were transferred easily. But that's not true today. We know that this paradigm is shifting very quickly. You can have enteric organisms that have DNA from distant archaea and distant eukaryotes. And so what we're faced with in the food supply is this idea of all bacteria live in a community. And we haven't really asked that question in that way. When we do food testing, we say, is E. coli here? Is Salmonella here? Is Listeria here? We ask it bug by bug. But in reality, this community is a huge community of bacteria are living together. And there's been some very nice studies in the last few years detailing in the gut, there's about 2,000 to 3,000 different microbes living together. And so there's plenty of opportunity to exchange DNA. So as the microbe changes its response, the host changes its response. And it's, good thing we all have an immune system, but it happens at the immune level. It also happens at the small molecule level, like organic acids. And so there's lots of people starting to think about spraying organic acids. So what does that mean for detecting the bacteria? It means we have a tougher job than we thought. And so the way we detect bacteria today is we go through this pre-enrichment phase. We collect a sample. It was just a big announcement this week that FDA's validated using 325 grams of a specific food. Well, for me, for you, that's probably not such a good sign, because now you have to use 4-liter containers to grow this thing in, and it has to sit there for 24 to 48 hours. And one company I know of built an entire new building just to house these 4-liter containers. So this pre-enrichment is really causing us grief broadly for a lot of reasons. And so this sampling and sitting around waiting for things to grow, where we have these rapid detection methods using PCR and genetics today, where we can do quantitative PCR, and the PCR assay itself takes only a few hours, but the enrichment takes a couple of days. And we believe that in the enrichment broth that it's growing the microbes that we hope it's growing. And typically that's not necessarily true. So by the end of the day, when you without using genomics, you're at about a 10-day detection phase when you start looking at typing and looking at exactly what the microbe is. And so the timeline across the bottom starts to become very long. So if we really want to focus how to get down detection times, we need to be upstream, look at ways to shorten the enrichment time or eliminate it possibly. So another way to look at this is, these are the steps that are happening today. And if we focus on just the early steps within the first 4 hours and ask the question, do we have the technological capabilities to shorten the detection time to less than half a day or less than a day? And the answer is we do. Technologically, we can do it today. The question is, do we have the will to implement it? Because we have to get out of the 1940s and 1920s technology and move into 2013 and start investing in and believing in more of a genetics and genomics world. So there's ways to capture, concentrate, and eliminate enrichment. There's ways to do a presumptive assay very quickly. There's ways to do genetics and genomics within hours. The question is, will we believe the results when we come back and have the answer? And so a lot of folks are believing that the answer to that is yes, including some of the federal agencies. And the way this is the reason it's happening is because next generation sequencing costs are so cheap. When I first started sequencing genomes 15 years ago, it was a 5-year, $350,000 effort. Today, literally today, it's about $75 in 4 hours. So it's cheaper than buying a nice bottle of wine to sequence a microbial genome. So now what do you do with it once you have it? You have this file that you can't open on your computer because the hard drive's not big enough. It doesn't fit into Excel, and you have to know how to program your computer to actually look at the data. So all of a sudden we have this paradigm that we're not sure how to use it, but it's possible to do this. And the reasons to do it are fairly compelling. If you look at the German outbreak of E. coli of 0104, it had a long lifespan of the outbreak because the genome was a combination of 2 other pathotypes that hadn't been seen in years. They got together, they recombined, they lost the EAE gene, which is the marker that we always use for PCR in E. coli. So now you had this variable result. You had a lot of false negatives, and the only reason they solved the German outbreak is because they sequenced the genome. Well, that was sort of not the first, but one of the first indicators that maybe this thing is going to be useful. FSMA allows new regulations and new reach for FDA overseas. How many of you heard about the tuna outbreak last year in Washington, D.C.? Nobody. A couple people. Relatively small outbreak, but the reason I point it out is because that tuna was processed by hand in India, shipped to the Middle East, put on a container ship, and shipped to the west coast of the U.S. and then shipped to the east coast of the U.S. And so by using genomics, FDA sequenced that organism. By using genomics, they tracked it back to exactly the person and exactly the processor in India. But it took them a few months to make that happen. So is there a benefit? I think so. I think we can shorten the time to do this. And that's where the 100K Genome Project comes in. And that's where the 100K project is sequencing 100,000 genomes. So what sort of context is that? In today, the number of genomes we have ready to use is 2,000. And we're going to sequence 100,000 in a few years. And we've been sequencing 2,000 for about 15 years. So some of my colleagues are saying there's no possible way you can sequence that many organisms that fast. And the answer is we can, because of the technology that's occurring today. So what do we do with it? What do we do with this database of genetic information around foodborne pathogens? So the FDA, NIH, CDC are going to put it into a public database, much like PulseNet. So PulseNet is a database of pulse field fingerprints, fantastic tool for outbreak detection and investigation. How many of you have run a pulse field gel? Painful, absolutely painful. But it's easy to digest because you have this limited number of fingerprint bands. There's, in Salmonella enteritidis, the pulse field pattern shifts by 1 band, which queries exactly 0.006% of the genome on 1 band, and they're making legal decisions on that amount of information. A pulse field pattern has 14 bands, approximately, say 25. The genome of E. coli or Salmonella has 5.2 million pieces of information on every genome. So the density of information that you're going to be using to make legal decisions, scientific decisions, is dramatically higher. And so we've formed this partnership, and the FDA is getting ready to implement this database called Genome Tracker. And it's intended to complement, replace, whatever word you want to use, PulseNet. And it's well on its way. Okay, so we've started this consortium of members in what I like to call an open consortium. So the founding members of this were my group at UC Davis and Agilent Technologies Food Group, and then CIFSAN. And we were all lamenting that we needed more genome information. And so we ended up getting together and putting this consortium in place, and we've recruited additional steering committee members, which include NIH, CDC, USDA, especially FSIS, and then a few companies are also involved at a level that they're really interested in pushing this forward. And we have these affiliate members where people are sending in isolates and sending in strains and sending in bits and pieces of things that we can use. And it's a representative of the U.S. through the Middle East, through Australia and Asia. And then there's a few corporate affiliates that we've brought on board who help us with special pricing, or they give us reagents, or they do something to help us push this project forward. Because all this information is going to end up in the public domain. And so it's not like it's going to sit in my laboratory, which I don't want, because I can't store it all. So it becomes a computer science problem on the other end of it. So we collect isolates from folks. When we agree that we're not going to send them anywhere, we sign a material transfer agreement. We isolate the DNA. We have metadata that comes with it. We sequence the genome, which I'll give a bit more information in a few minutes. And then at the end of the day, that information is going to be publicly available for people to go mine it and do what they want with it to find these genetic features that they're interested in. And then all of that's going to end up in NCBI. So the isolate submission is fairly simple, in my opinion. We do this material transfer agreement. We agree we don't do anything nefarious with your organism. We won't give it to anybody. If somebody wants it, then we send them to you. And then you can decide if you want them to have it. We authenticate the organism when it comes in, and we've had a few troubles. There's been a few that have been contaminated. We bank it, isolate the DNA, and then at the end of it, we return the sequence back to you. And if you want to collaborate, we're happy to do that. You have 12 months moratorium on what you want to do with the information. After that 12 months, then the sequence is released into the public database called NCBI. Okay? So we're trying to be very fair about if you have some very specific information that you want out of those genomes, we give you plenty of chance. 12 months is a long time to mine the data. Okay? The organisms that we're focusing on first are the ones at the top of the list, and they're the ones, of course, that are the most prevalent in foodborne outbreaks. We've also got a collaboration with norovirus and hepatitis A with a group in the Netherlands, and we're trying to get something going with a group in Georgia as well. And so we're trying very hard to get a worldwide representation of isolates. And I know this is really hard to see. I apologize. The big blue one is Salmonella. We've got about 3,500 isolates banked and authenticated. But the biggest part of those are Salmonella, and the next biggest is Vibrio. And then pending, sitting in our freezers that we haven't gotten to are another 15,000 isolates. And as soon as we can figure out our permitting issues with USDA, which I think we're very close, this is going to go to 90,000. And so we've got one group who wants to contribute about 90,000 isolates all in one shot. So for those of you who grow bacteria, think about how much effort it's going to be to grow 90,000 microbes and bank them and authenticate them. But what's interesting to me is the bank is shifting to Campylobacter. Almost everybody is interested in Campylobacter. And I think partly because it's so difficult to grow, it makes it a logical target for genetic tools. The representation today is we've got about 75%-ish of the isolates are coming from North America. Reasonable, but considering the amount of food that we import, we need to have a broader vision of what's going on around the world. And so we're working hard to find some collaborators in Africa, Asia, and India as well. We've got a few, but not quite as many as we're hoping for. So just very quickly, this is sort of the process that we're taking for getting this to work, and that is the banking and the isolating the DNA is the easiest part. Then you have to do this library construction. So you take the DNA, you shear it up, you put it into a molecular library. And we're using three different techniques to sequence. Not all at the same time on every organism. Depends on which organism, but those techniques are going to be used. And then the databases are created and the data go back to the submitters. So we're nearing the end of the first year of this project. And where do we stand? We stand at we're focusing on Salmonella at the top 50 most common serotypes. We've got a variety of worldwide partnerships developing that are coming along very nicely. We'd like to develop a few more partnerships. And we're ready to sequence 1,800 of the 100,000 organisms, which is what we promised FDA we would do in year 1. In year 2, everything will be in place. The automation is in place. We'll be sequencing 25,000 isolates per year for the next few years. And that's a very doable number, especially at the cost we're talking about. And so we've already, in the first year, we've got 90 sequenced now, and we've done it with 3 technologies, and we've already found some really, really interesting things that no one's ever seen before in only 90 isolates. And that's mostly with Listeria and Vibrio. So as we start doing more and more and more, there's going to be all this phenomenal science to be done. But what are we going to do with it from a food safety perspective? And that is, sort of the challenge of the next phase. And the people who are interested in submitting the isolates. We have a website that you can go check out. You can contact me initially, and then I'll get you to the right person if you want to submit some isolates. I'm getting ready to do a major upgrade on the website so you can track what's going on, who's done what, where are we in the process. With the whole idea of actually bringing, how much time do I have? Bringing this to a place where people can use it. 15 minutes? Okay, I'm good. People can use it for really doing public health outcomes, and that's the goal of the FDA and the USDA for sure. So when we have these steering committee calls, which we do monthly, you get FDA, CDC, and USDA on the telephone. It's a very interesting dynamic among those agencies and what they want to do with genomics and how they're implementing it and what they think they're going to do next. It's very clear FDA is already using this in outbreak investigations. And they're working on ways to make it faster. And they're working on ways to what's the next phase of this detection piece. Are we going to make things growth independent, which CDC is very adamantly opposing. If you don't have an isolate, you have a hard time doing something next. Yet we have the technology today. We don't need an isolate. We can go forward and have the genetic signature. And we can start looking at things like virulence and antibiotic resistance. There's ways to look for fingerprints of the genome of where it came from. So just like humans, we have fingerprints of where we came from, so do bacteria. So it's bringing up this whole topic again of culture-independent methods in the food supply. In the past, as a group, we've not been very willing to invest in culture-independent methods, in part because of BAM and in part because of FDA's mandate that we had to grow everything. It seems to me that they're softening that topic. And so now we're starting to be able to come to these newer rapid methods that are truly rapid. I was involved in a lot of DoD research in the early 2000s, and rapid methods to them meant minutes. Well, rapid methods to the food supply means less than a week. There are technologies today that can get at minutes in the food supply. We haven't been willing to invest in those or think about them in the past, but we certainly are approaching that again today. So I want to give you a couple of examples about what we can do with some of these techniques. And a lot of these are going to be foreign to what you're used to seeing. There's lots of molecular biology that can be done, and we can start comparing genomes, and we can use SNPs. And this is how the FDA is using things today, is they look for these single nucleotide polymorphisms, and they make these trees again. And then they use the trees to say, is it in the outbreak or is it out of the outbreak? Is it in the farm, not in this farm, or in this environment? And there's other things that we can do to look at forensics, looking at the genome content. But what's being done today is the SNP analysis. And so I'm going to skip a couple of these. So I want to give you a perspective of something that's in place today that can be used today. It was developed for DoD a long time ago. And so there's this fluidized bed capture concentrator that you can run large volumes through. Entire 4-liter containers will go through this. And you do it in about 15 minutes. And then you do an ELISA, which you're used to doing, the dipstick tests. And you get a presumptive ID within about 35 minutes. Well, if you then take those beads and you extract the DNA or extract the RNA, you can start using that for molecular serotyping or molecular analysis broadly. And all of that can be done in about 3 hours. And so a while ago we examined what it looks like for this rapid concentrator compared to commercial assays. And we did it in hamburger, because you can take 325 grams of hamburger, put it in the enrichment broth, and run the whole slurry through this concentrator and capture the bacteria, E. coli, from it. And we did it in hamburger, and you got about an 80% positive if you did it directly from the food, 40% positive if you took the enrichment broth, and then that matched up with the commercial assay of about 30% positive. So when I first showed this data a long time ago to the food industry, nobody wanted to talk to me, because now I'm going to start finding more positives. I'm okay with that. However, the food safety problem ain't going away. So what are we going to do about it? We either have to find the real positives and do something about it, or we're going to be in the same boat that we're in today, and FDA is going to have 5 million pieces of information to come and talk to you about. So it's in this paradigm now where they're going to have this huge body of information. And so it's going to be very diagnostic. So I stopped eating bean sprouts a long time ago because I couldn't find any that didn't have E. coli in them. And that has no indictment on the sprout industry whatsoever. It just is a reality that E. coli is associated with bean sprouts. And we did the same thing with apple juice, and we started using receptors that bacteria bind to specifically when they invade. And so we wanted to give an idea of what are the more virulent organisms that we want to see. And we did it in milk and salami and so on. But the detection limit is very small. We can make this be really sensitive, less than 10 cells, in 4 liters. Okay? Or we can make it less sensitive. And when you have organisms that are zero tolerance, now you have what next generation or whole genome sequencing is WGS. Now you have this tolerance level means a whole new thing because you've got genetic fingerprints in this genetic mixture, this community, that they can find, we can find. We don't need a colony. And so these sorts of assays are very robust, very small variation. And so we've been working on another assay that's very quick and easy to look at serotyping at the moment. And so we can do molecular serotyping in a couple of hours, and we started using RNA instead of DNA. And so we don't need to, if we use RNA, we know the microbe's alive, we know it's growing, we know it's expressing genes. There's an argument among many people that you can have dead DNA and you'll find it. Not in my hands, not in food. If the DNA is there, it's not naked out in the environment. It's not from a dead microbe. It might be from a microbe that you can't grow, but it ain't dead. So, and we just have a paper in review right now showing that that's the case, and there's been 5 or 6 other papers published in the dairy area showing that if you find DNA, it's from a microbe that you can't grow. It's not from a dead microbe. Okay. So anyway, this happens very quickly. We can extract the DNA. We validated it with nearly just over 1,500 isolates, with the most 40 common. We've done it in multiple matrices. And it's been used in 3 independent labs, and we're 100% accurate for telling you if you have salmonella. And we're about 98% accurate to tell you what the correct serotype is. So within 2 hours, you can have the serotype of the microbe using DNA or RNA, and you don't have to go through the long, laborious, however many weeks it takes. So how are we going to implement all of this? It's very difficult, because now you're hearing about all these tools and techniques that you're not necessarily geared up for. The government is starting to use them. And this is a case where FDA has actually truly been a thought leader and has really gone to the next step and said, we're going to embrace this next generation sequencing because we can harness the capabilities of this sequencing and we can do something about it for the public benefit. So that's been a little scary for processors and producers, because now we're challenging the techniques that we've been using for so long and that were developed in the 20s, 30s, and 40s, and we're very comfortable with. So so now we also have a workforce issue. How many people in your company or at your place know how to do this? Hardly anybody. So we need this all the way from veterinary care to animal husbandry to food science to food safety to now adopt this approach and thought process about how to use next generation sequencing. And I used to be in a food science department, and I can tell you we never talked about genomics except in the research lab. We weren't teaching our students about how are we going to use genomics in food safety and how are we going to use genomics in the next phase. And I know you've heard a bit in the meeting about using genetics for animal approaches, and it's being done. We can do it on the microbe as well. So the benefits are faster assays that are much more specific. And so what you're faced with today is sort of this middle bar of the colony enrichment multiplex PCR assay that is hit and miss. So the idea of PCR, we have had less robust PCR assays because we don't know the genetic diversity of that particular gene. Like EAE, where it can be lost, where it can acquire new mutations. And so people have tried to do multiplex PCR. Well, that's even harder. But the base problem really results in the evolution of specific genes. So the fewer genes you use, the more likely it is that you're going to be, quote, wrong, or you'll get a false negative. The culture-independent methodologies give you a fast time frame, give you something that you've known with techniques that you're used to seeing and trust, mixed with some other newer techniques. And then on the far right is the next generation sequencing, where you do everything by sequencing. And the paradigm becomes very doable. We can do this in the lab today. It's almost routine. We've got it to the point where we can automate it. So it will be very routine, and it's less than a few days of getting an answer. Okay. So that's one of the reasons that the federal agencies were so excited to do the 100K, is because now you'll have enough genetic information to look at the diversity of genes, and you'll be able to create more robust PCR assays. So I have enough time. I want to give you, right, I have 5 minutes? 4. I'll give you one quick example of how we use this in a salmonella outbreak in sea mammals, and I won't go through the whole thing. So each of these bars represents a specific tissue in a series of outbreaks that occurred over about 12 years. And it's a Salmonella enteritidis outbreak in sea mammals. And so this is the total number of SNPs, or total number of genetic mutations across these individual genomes that we were analyzing. And so the way to read this is each of the rows is a specific spot in the genome, and it told you, did it change, did it have a SNP, did it change the sequence? And these are 3 tissues, and the isolates look very similar. And then we looked at sea mammals across 98 to 2008, and all of a sudden you get great big blocks of changes. So how did you go from what looks very similar in 93 to something looking very different in those out years? And when you start looking at rodent and equine and other sea animals, you start seeing these intermediary shifts. And so very quickly you can watch the evolution of an outbreak, even over a long frame or a short frame. And that's with only 500 pieces of information. So you can really start using this in a very powerful way. I use this example in sea mammals. This is exactly how FDA is using it for foodborne outbreaks. And they create these trees again, so you can look at it.